Hello, everyone, and thank you for taking the time to join us today for this exciting webinar being brought to you by Syntica. My name is Jade, and I'll be your host for today's webinar. So today, our presentation is entitled NIR-II Fluorescence Imaging, Why, When, and How. Today's webinar will be given by Alexi Francis, who is an accomplished international sales manager with over 8 years of expertise at Vilber Bio Imaging, specializing in preclinical optical imaging. Throughout his career, Alexi has successfully brought cutting-edge in vivo imaging solutions to research development teams across the globe. His understanding of preclinical imaging technologies, coupled with his expertise in optical fields, has elevated the development of the Newton 7.0 series to new heights. Now, before we begin, I'd like to briefly mention a few housekeeping items. We anticipate that the presentation will be about 45 minutes, which should leave us some time for questions. Please submit your questions in the Q&A dialog box throughout the presentation, and we will go through them at the end. We'll also create a transcript of questions that we do not answer during this session, as well as those that we may not get to, and this will be made available after today. Please also note that we're recording today's session, so if for some reason you lose connection or can't hear everything clearly, you'll have access to the video file afterwards. Now, without further ado, I'm pleased to turn things over to Alexi for today's webinar.
Thank you, Jade. Hi, everyone. Thank you for joining this webinar. I am pleased to give you some more information about NIR-II fluorescence imaging today with the use of our Newton 7 FT900 that I will present in details. Just before starting about the applications and more scientific discussion, we'll start with a presentation of the company first, Vilber, so that you have a better background of where is Vilber coming from, what we're doing, so that after that presentation, we can go over the technical discussion. So Vilber is a leading life science company whose headquarters and manufacturing plant are based near Paris in France, with regional offices in Germany, China, as well as Singapore. So Vilber has a presence in over 100 countries through a network of established distributors such as Syntica in North America and a few countries in Europe. And we have equipped so far more than 20,000 laboratories around the globe. So Vilber was created in 1954 and started as a small startup company manufacturing UV instruments such as UV cabinets, DNA crosslinkers, radiometers, as well as UV lamps. And then it's from the 1980s that Vilber started imaging systems with the release of gel dock instruments like the dock print for DNA RNA gel imaging. And 2002 was a transitional year for Vilber, who were the first manufacturer to equip imaging systems with a fixed lens for chemiluminescence detection on Western blots, having the widest lens aperture to collect the maximum amount of light and reach unrivaled levels of sensitivity. In 2011, we started to receive some publications with images of small animals taken with our Western blot imagers like the Fusion FX7, and that was providing already a great sensitivity for small animal imaging with bioluminescence activities in vivo. So that's how the idea of a proper instrument dedicated to small animal imaging emerged, releasing the Newton 7 in 2016, which would include animal management accessories like a heated bed and nose cones being compatible with any anesthesia stations. But we didn't want to stop there, as we wanted to provide our users a system that would push the limits of a 2D imager, featuring the possibility to better understand deeper tissue structures. Thus, the launch of the Newton 7 FT500 with a comprehensive 3D optical tomography module. So again, that's not where we stopped. We wanted to upgrade again the possibilities of optical imaging. And last year, we have released another instrument, the FT900, with a second camera dedicated to SWIR or NIR-II, which I will detail in the next slides. So now we can enter the Newton 7 presentation. I'll start with the fluorescence applications. So I'll start from the scratch with the concept of fluorescence in vivo imaging. Then we'll head over the SWIR and NIR-II fluorescence imaging, which is kind of a new trend for optical imaging. And we'll head over the bioluminescence capabilities of the NIR-II. We'll go through some case studies, technical presentation about hardware, but also a presentation of the software. So you have a comprehensive background of the instrument. So Vilber is proud to release its brand new optical imaging system for in vivo, in vitro, and ex vivo imaging application, the NIR-II FT900, dedicated to bioluminescence as a 2D option, but also with a 3D bioluminescence tomography feature and a visible NIR-I, but also SWIR NIR-II fluorescence applications. So let's start with the fluorescence imaging concepts. Fluorescence preclinical imaging in vivo is a technique used in biomedical research to visualize and track biological processes in living organisms using fluorescent markers. So molecules called fluorophores are used. These could be fluorophores like dyes or fluorescent proteins or quantum dots. And in regards to the applications, it is widely used in cancer research to track tumor growth and metastasis using fluorescently labeled cancer cells. It could be used in drug development, assessing the biodistribution of drugs, evaluating treatment response, infection disease to monitor the spread of infection using fluorescently labeled bacteria or viruses. And you can study also the gene expression using fluorescent reporter genes to study gene activation in different tissues. So the advantages of fluorescence imaging is that it's a non-invasive technique which allows longitudinal studies, meaning the same animal can be imaged multiple times. It's pretty high sensitive because the signals can be highly sensitive, enabling the detection of even small number of labeled cells. And you have a real-time monitoring, which provides dynamic information about biological processes as they happen. There are some limitations, though. The fluorescence signals could be attenuated by the tissue, limiting the depth of imaging, which is typically effective for superficial tissues within small animals. The autofluorescence could be also another issue. Some tissues naturally emit fluorescence, which could interfere with signal detection. And the quantification could be altered since the signal intensity can be influenced by various factors like tissue absorption and scattering, making quantitative analysis challenges. So in regards to the strongest limitation is the background of autofluorescence, since many biological tissues like the skin exhibit autofluorescence, especially excited by visible light. So here we have some images of negative mouse, just to show you the differences when we excite the animals at different wavelengths. So for instance, here at 480 nanometers, we can see that there's a strong autofluorescence. So at 480 nanometers, we have to illuminate the animals with blue light. And this blue light is being reflected onto the animal skin or the animal fur, which creates a very strong autofluorescence, with which it's very difficult to distinguish the true signal from this autofluorescence background. In the red spectrum, around 640 nanometers, this autofluorescence is reduced, but still quite present. And when we excite or illuminate in the near infrared, around 780 nanometers, this autofluorescence is drastically reduced. So the fluorescence signals in the NIR region from 700 to 900 nanometers can penetrate deeper and are observed to travel more efficiently through the tissues than ones in the visible light spectrum. So that's where the near-infrared spectrum comes into place. It's to attenuate the autofluorescence background so that you can detect deeper signals within the animals. Nonetheless, the instrument is compatible with all the most commonly used fluorescent dyes in the market for small animal imaging with fluorescence. So here we have 8 excitation channels from 400 nanometers to 800 nanometers. This is for a traditional visible and NIR-I fluorescence imaging. And having multiple excitation channels helps to also monitor multispectral imaging. If you have a mouse labeled with 2 types or 3 or even more kind of markers within the animal, it's also possible to distinguish all those markers in the same subject if the markers are properly separated by at least 100 nanometers apart from each other. So here on the image, we have, for instance, subQ tumor carrying a NIR fluorescence, another subQ tumor with GFP. So here we could clearly distinguish the two different tumors. So the instrument is scanning the animals with different profiles, and then all the signals are overlaid over the visible image so that they can be distinguished visually on the single image. So we can go over a case study from a French pharmaceutical company called Nanobiotics Curading. And here they are working in nanomedicines. And the aim of their study is to propose a new approach to change the way nanomedicines are biodistributed by priming the body before receiving the treatment. So for that, they have redefined the nanomedicine's bioavailability with a liposomal nanoprimer having specific physicochemical attributes. So they're using a homemade fluorophore having an excitation at 640 nanometers and an emission at 700 nanometers. And this nanoprimer solution was injected in the tail vein of the animals. And then the fluorescence acquisitions were performed on the whole mice for 1 hour inside the instrument. So they have left the animals for 60 minutes, capturing an image every minute. So it's a biodistribution study. And here we can see the different time points after 5, 10, 20, 30, and 60 minutes. And within 10 minutes following the injection, we can see that most of the product was being accumulated in the liver, with only a small fraction remaining in the blood. So when we do a serial acquisition like this one, at the end, you have the option to translate all those captured images into a time-lapse video. So that's very useful for a video presentation, for instance, to show your work and to better understand the distribution of the drugs within the animal over the period of time that you have imaged the animal. So that was the traditional visible and NIR-I fluorescence imaging. Now let's head over the SWIR or NIR-II imaging. SWIR refers to shortwave infrared, also called NIR-II, since it's the second spectrum of the near-infrared, and it operates in the 900 to 2000 nanometers wavelength range and has gained considerable attention for in vivo biomedical applications. SWIR, or NIR-II, offers several advantages over visible and NIR imaging due to its unique optical properties that I will detail in the next slides. So SWIR imaging is, as we saw, above 900 nanometers to 2000 nanometers, and the SWIR light can penetrate biological tissues much deeper than visible light. This is because SWIR wavelengths experience lower scattering in biological tissues, allowing light to travel further before being scattered or absorbed. So we also have other advantages such as a reduced autofluorescence. So the biological tissues such as the skin or the fur is strongly attenuated. So this background signal can usually interfere with the detection of label fluorescence, but in the SWIR regions, autofluorescence is minimal, resulting in a much higher signal-to-noise ratio, which leads to clearer images and better contrast, especially important when trying to detect small signals or rare events in vivo. So there's a better signal-to-noise ratio due to the lower scattering and reduced autofluorescence, and the emitted SWIR signal from the fluorescent probes or the contrast agents are less distorted and more distinct. This higher signal-to-noise ratio is crucial for accurately tracking biological processes such as drug distribution or the movement of specific cell populations within the body. There's also an improved image resolution and clarity. Although the resolution generally decreases with longer wavelengths due to the diffraction, this reduced scattering in SWIR compensates for this effect in biological tissues. As a result, SWIR imaging often provides sharper and clearer images compared to visible light imaging when imaging deeper structures. We have also as an advantage a reduced light absorption by biological chromophores, and the SWIR light is less absorbed by these chromophores, allowing it to travel deeper into the tissues and enabling better visualization of the internal structures. We have an enhanced imaging of blood and vascular structures. So it's highly sensitive to the absorption properties of water and lipid content in tissues, making it especially effective for imaging blood vessels and other vasculature structures in vivo. So this is a valuable tool for the microcirculation detection. Other application could be tumor imaging, detecting deep-seated tumors and monitoring their growth or response to therapy. Drug delivery to track the biodistribution of nanoparticles and drug carriers, which can be labeled with SWIR-emitting fluorophores. We can pay attention to brain imaging, which is a non-invasive imaging of brain activity and blood flow in small animal models, even through the skull. You have access to circulatory and lymphatic system imaging to visualize blood vessels, lymph nodes, and lymphatic flow. And also the main advantage of the Newton-7 FT900 is that you can also co-register bioluminescent tumors with their fluorescent vascularization. Since we have two cameras in the instrument, you can first capture the bioluminescent tumor, and then you can visualize its vascularization using the SWIR detector. So here we have a small case study from, so we have some images here coming from the company NIR, for which we have tested their fluorescent probes in the NIR-II spectrum. So here we're looking at NIR-I and NIR-II fluorescence images obtained on mice carrying a Core33 type sub-Q tumor expressing EGFR, which is the positive tumor on the left of the animal, and also a small HCC 1954 tumors that express very little EGFR, which is the negative tumor on the right of the animal. So it's the smaller tumor. Those were 24 hours post intravenous injection of the Wasabi 1000 panitumumab from the company Fluo NIR from France, which are soon commercializing those NIR-II probes. So here the EGFR being the growth factor receptor and the panitumumab, the antibody that targets it. So for the same exposure time of 100 milliseconds with the NIR-I and NIR-II, we can see that NIR-II provides more sensitivity, more depth detection, but also sharper signal patterns. So the conclusion of this comparison from NIR-I to NIR-II is that when using NIR-II, we could detect smaller lesions. So, excuse me. So we could detect smaller lesions, which should be more easily detected with the NIR-II rather than the NIR-I. So it provides better sensitivity, more depth detection, and sharper signal patterns. So at the conclusion, SWIR imaging has a great potential for in vivo biomedical applications due to its ability to penetrate deeper into tissues, reduce scattering, and provide clearer images. It opens new possibilities for non-invasive diagnostics, real-time imaging during surgery, and the study of complex biological processes. Despite its challenges, ongoing advancements in imaging technology, such as contrast agents and clinical adoption, could make SWIR or NIR-II a valuable tool for medical diagnostics and research in the near future. So that's the NIR-II part, and we'll head over the bioluminescence activities of the Newton-7. So for bioluminescence, we usually use genetically modified animals that are expressing luciferase cells, for instance. So here we're looking at some images coming from Antwerp University, from Belgium, where they have injected 500 cells in mouse A in the left and 50,000 tumor luciferase expressing cells in the brain of the animals on the mouse B on the right. And they were those cells were injected 1 week before the imaging. Then the day of the imaging, the researchers have injected the deluciferin substrate and waited about 10 minutes before capturing the images. So mouse A with 500 cells needed 5 minutes of exposure time, and mouse B with 50,000 cells needed 1 minute of exposure. And here we can see the great sensitivity of the Newton-7, since 500 cells is a very low number of cells, especially in the brain, and especially when it was injected 1 week before the imaging. So usually They had to wait about 3 weeks before starting to see their signals with their former imager. Now with the Newton, after only a week, they could start their study because they can already visualize the first signals from the brain activities. So that's the traditional 2D bioluminescence imaging. The Newton-7 also offers the capabilities of 3D optical tomography for bioluminescence detections, which provides more details about the signal size and its localization within the animal body. So the 3D bioluminescence tomography is a recent technique that enhances the traditional planar 2D bioluminescence imaging into a quantitable 3D approach. And when we see the 2D image on the left, we can see the signal that is being represented on the surface of the animal. But there we have no information about the depth of the signal. So we have a lack of depth perception, which leads to approximate results, which could be under or overestimated, so that the data can be misinterpreted. So the 3D tomography overcomes this challenge as the signal is being reconstructed into a volume and repositioned within the 3D representation of the animal, which gives clear details on the signal size and its localization within the animal body. Here we can see some bones and organs. Those have been digitalized, so it's an atlas which is accessible from the software, and it's more to be used as a tool to better understand where is your signal located within the animal. So what you have as a concrete information and data is the 3D mouse surface, as well as the signal repositioning and the possibility to quantify that signal as a volume in cubic millimeters and surface in square millimeters, so that you can monitor the progression of your tumors or metastasis over time using the 3D tomography. So how do we achieve this 3D tomography tool? So first, the topographic image of the mouse is being captured with the help of 3D NIR cameras on each side of the darkroom. So those cameras are being used to calculate the 3D mouse representation. And then the main CCD camera from the top is acquiring multiple images of the signal using various emission filters, depending on the spectral emission of the luciferase properties. And eventually, the signal is reconstructed with the use of an established algorithm and repositioned within the topographic image of the animal. So as a result, we have a much better understanding of the signal's depth, and you can quantify it as volume and surface. So here we have a case study and images coming from Coimbra University in Portugal. So here we see the 2D image with the lung tumors, and activating the 3D tomography module, you can then quantify those signals and better understand the depth of those signals. But here we knew that it was coming from the lungs.
All right, awesome. Thanks, Alexi, for the presentation so far. We'd like to take a quick break and ask the audience to tell us a bit more about the research that they're currently doing. So we've got four poll questions prepared that you should see pop up on your screen. So the first one, it's asking if you've used in vivo optical imaging before. So that would be your bioluminescence or fluorescence. So your options, yes, currently using it. Yes, and I've used it in the past. No, but I hope to add it to my optical imaging capabilities. Nope, just here for my own scientific discovery and curiosity. Second question, what's your primary reason for attending this webinar? So your options are to learn about the benefits of NIR-II imaging over your traditional visible NIR-I imaging, to explore specific applications of SWIR imaging, to gain a technical overview of the Newton FT900, and then to network with others that are in the field. The third question, what applications are you most interested in for the NIR-II imaging? So we're looking at vascular blood flow analysis, tumor targeting and angiogenesis, drug dispersion and metabolic studies, brain and single cell imaging, and then others. So we welcome you guys to specify so in the chat. And then the last question, which conferences do you typically attend? So this is a multi-choice answer, so you can select all of the ones that apply. So American Physiological Society, American Association for Cancer Research, the European Association for Cancer Research, World Molecular Imaging Congress, and others. So again, please use the chat feature there if you want to just drop the ones that you typically go to. Perfect. All right, great. Seeing some good answers roll in there. Perfect. Awesome. All right. Thanks everyone for telling us a little bit more about yourselves. I'm going to turn things back over now to Alexi.
Thanks, Jade. Right, so now let's carry on with some more information, technical information about the Newton-7 FT900. So we have one instrument, one imaging system with two cameras. So there will be our well-known DOC-Q11 camera used for 2D bioluminescence, but also 3D bioluminescence tomography and 2D visible NIR-I fluorescence. Yeah, now we have to distinguish NIR-I to NIR-II. So the DOC-Q11 is for bioluminescence and visible NIR-I fluorescence, while we have a dedicated camera for SWIR detection. So that's the second camera. And those two cameras are being interchanged whenever you're selecting the profile you want to use. So the mice stay in place on the heating stage. And whether you select bioluminescence or SWIR, the two cameras are motorized and can alternate to each other whenever you need to switch the application. So that's how it's working, so that you can monitor and combine bioluminescence activities over fluorescence SWIR activities. So in the Newton-7 FT900, we have those two cameras, which are both equipped with our f/0.70 lens aperture for maximum sensitivity. So both cameras have their own lens, which is 0.7 aperture. And we have 10 excitation channels from 440 nanometers to 980 nanometers. So we have added two additional light sources at 880 nanometers, as well as 980 nanometers. And we have 14 high efficiency emission filters spanning from 500 to 1600 nanometers. So I would say the traditional visible NIR-I fluorescence stops at around 850 nanometers for the emission filters. Then we have other emission filters for the SWIR region with 1100, 1200, 1300, 1400, 15, and 1600. The NIR-II camera has a 25 frame rate, which is ideal to study real-time metabolism, such as drug distribution, heart rate, and breathing. And both cameras are they benefit from an absolute calibration with NIST certification. So that's the FT900 on the bench. So you can see it's quite compact. It's benchtop, which is very useful to install the instrument anywhere in your lab. There is no radiation in the FT900, so that you can install it anywhere you want. Inside, you have a sliding out heated stage with a platform here for 5 animals with 5 nose cones that you can individually turn on or off according to the number of mice to image. So here we're looking at a phantom mouse. And from the software interface, then you can select the field of view of your choice. So here we have a 5 mice position, for instance. You have options for 4 mice, 3 mice, 2 mice, 1 mouse, or a macro imaging mode. So in the video, we go to macro mode. So both cameras are motorized on the Z-axis. so that you can bring the cameras very close to the subjects. That's ideal for study of brain activities, for instance, especially using SWIR. And the platform, the mouse stage, is also motorized on the X and Y axis. So you can see on the top right corner, you have some up and down arrows and green, left or right arrows. So you can move the stage to bring the subject on the top or on the left or the right. If you prefer to see a lower part of the body, it's still possible to move the stage so that it's the lower part of the body is in the field of view. So once you have selected your field of view, you simply select the profile, the applications you want to use, and you press start. So we have 3 acquisition modes. The auto mode, where there will be an automatic exposure time that will be calculated in regards to the stronger signals, so that the camera can capture a maximum amount of data without reaching the saturation. Then you have a manual mode where you simply enter the exposure time of your choice. And there's a serial mode where you can launch a series of images to be captured. So you select the number of images to be captured, the exposure time for each image, and the waiting time between each image. And then you press start. So that's very useful for biodistribution studies or pharmacokinetic studies. So here we press start, and we'll see some nice signals of brain tumors from bioluminescence activities. So for fluorescence imaging, we have 2 panels of LEDs from 400 to 900 nanometers that offers a unique scanning technology to provide homogeneous illumination all over the stage surface. And this makes sure that each positions receive the exact same amount of light intensity. So if you have mice on the sides and mice in the middle, we make sure that each mouse receives the exact same amount of light intensity for an accurate fluorescence quantification. For SWIR imaging, we have added 2 additional light sources at 880 nanometers and 980 nanometers, which are fixed. So those light sources are not scanning as we see now, but they are fixed so that you can do the biodistribution study with the real-time imaging from the 25 frames per second capabilities of the SWIR camera. And both cameras are equipped with our widest lens aperture f/07, which provides enhanced sensitivity, faster and more light collection down to single cell imaging capabilities. So we not only have the FT900 that I just presented, we also have other configurations to suit your needs, your imaging needs. So we have the FT100. This one is even more compact and is dedicated to bioluminescence with 2D, 3D tomography, as well as visible and NIR-I fluorescence. It's dedicated to 3 animals, so that's why it's more compact. Then we have the FT500 with the capabilities of 5 to 10 mice, and the FT900 with the second camera for SWIR applications. And for those of you who don't need bioluminescence or the visible fluorescence, we also have the NIR-II only instrument called IR500. So that system will be only equipped with the NIR-II camera. So that's all the configurations we have for the Newton 7, FT100, FT500, 900, and IR500. We also offer a comprehensive range of accessories from the anesthesia station called the biosthesia, which is made of an oxygen flow meter, an isoflurane Take-3 vaporizer. You have an induction chamber, dual delivery manifolds to deliver the gas to the induction chamber as well to the imager. And there's a waste gas scavenger, which traps the excess of isoflurane into charcoal filters. We have also designed an isolation chamber, which is an SPF chamber sealed with HEPA filters. And so it's a chamber in which you can use to transport the animals, to anesthetize them, and to image them without having to opening the lid. So the lid is made of a crystal clear quartz. The base is heated to plus 37 degrees Celsius. You also have individual on and off manifolds to deliver the gas. And this isolation chamber is also compatible with our external docking station. So the docking station can be connected to your anesthesia station here. And when you connect the isolation chamber or the other animal stages, you can do some preparative work on the animals. You can do some injections. And then when you're ready, so the mice are maintained under anesthesia. And when you're ready to image, you simply disconnect from the docking station. You connect inside the imager and it's ready to image. So you could also have multiple stages. So we have stages for 10 mice, for instance, or 3 rats, or you could use 2 stages for 5 mice. So while a batch of 5 mice are being imaged, you could prepare the second batch on the docking station. And whenever the capture is over, you can simply interchange the 2. So yeah, you have less preparative work and it's a gain of time, mostly when you have 100 of animals to image a day. Now we'll have a look at the analysis software called Quant that we have developed in-house. So the Quant analysis software is a license-free software with an intuitive interface, which is very user-friendly and ready to export data. So with the Quant software, you have the possibility to display some regions of interest of your choice. You also have an automatic detection of all the regions of interest, all the signals, and you can visualize your signals as 3D representations as well. So the image you see on the bottom, so which gives you a nice look over the different signal concentrations within your image. So you have adjustable scales that you can switch from counts to radians. And when you're ready, you can also do some multi-view analysis. So you can load unlimited number of images where you have the possibility to display regions of interest automatically over all the images that you have loaded with a one-click scale extension, one-click RIs applications, and a one-click quantification. So once all your images have been selected, then you can go over the table and export this table to Excel, which shows you your table and the data either in counts or radians units. And also you have other sheets with the images and all the acquisition settings from the capture. You also have the possibility to export your image and results as a presentation. So we have reports that can be generated as a PDF or as a Word document before being saved as a PDF or to be brought to PowerPoint for your presentations with all the information about the image capture, and it's every regions of interest individually have all the information about the counts and the radians that you can show in your report for the presentation. So I think that's pretty much it for the FT900 presentation and the NIR-II information. Thank you so much. I think the next step is the Q&A session, so you can enter your questions if you have some.
Perfect. Thanks, Lexi, for the great presentation. So yeah, as he mentioned, we're going to take some time now to go through some questions. Again, please feel free to drop them there into the Q&A dialog box. I can see already we had one question that popped in a little while ago. So we have Carlton asking, can you see superficial tumors when the mouse is flipped over? So essentially they're asking if you can see through the entire mouse and essentially, what is the depth of penetration for the NIR-II?
I would say yes, you could see it, or also that's not necessary to flip it over if the signal is on the other side. So it's best to position the mouse in the imager where you expect the signal to see, either on a ventral view or a dorsal view. So if you expect the signal to be on the in the organs such as the liver, for instance, you can place the animal as a ventral view to see the belly. And then when you expect the signal to be more on the back, we call that a dorsal view. So, but if you flip the mouse upside down, yes, because the tissue becomes more transparent, you will see a little bit of signals for sure, but not the best. That will not be the best option.
Just quickly then on that, is there a sort of threshold then that the NIR-II can actually penetrate down to? 1 centimeter or more or less? Is there an actual threshold that you guys would say?
So traditionally with NIR-I penetration, we can go through down to 0.5 centimeter. So I would say that with an NIR-II, we can go much deeper after 5 millimeters down to 10 millimeters. Yes.
Okay, perfect. We have another question from Mark. Can you let us know what the illumination intensity is with the 980 nanometer illumination?
You can select the intensity, and for 980 nanometers illumination, it's from 100 to 200 microwatts.
And then another question from Mark. What is the longest practical exposure time that can be used with your NIR-II camera? Is it cooled? We can correspond offline if it's easier to discuss those details.
Sure, I'm happy to discuss later on, but to answer your question, yes, the camera is cooled to minus 60 degrees Celsius, whereas the other camera for bioluminescence is cooled to minus 90 degrees Celsius, absolute cooling. So the in-gas camera has a minus 60 Celsius cooling. And the question, the longest practical exposure time that can be used. So usually you could go over a few minutes, especially if you want to see the distribution of some nanoparticles or other treatments within the animal. So usually, yes, 5 minutes should be fine, or you could go to longer exposure if needed.
Great. Another question from Jan. So essentially asking us, what is the size capabilities of the system? So could a rabbit fit inside? Would you be able to image superficial organs such as the trachea?
So a rabbit could fit inside the instrument, although depending on the size of the rabbit, it may not fit in the whole field of view. So we have recently installed an instrument, a Newton-7, to image rabbits, and we have custom made a stage for a rabbit actually, so that it can be laying on its back to visualize the leg of the rabbit. So we can adapt the Newton-7 to other small animals such as rabbits, guinea pigs, and but most of the studies are done on mice.
Awesome. We also had a couple of other questions that were submitted in advance. So what are there any potential challenges that researchers could face when transitioning from NIR-I imaging to NIR-II? So I'm thinking, availability of fluorophores, et cetera. And then how does the Newton 7 FT900 and Vilber kind of simplify this process or offer support that way?
So we are still at the beginning of the NIR-II fluorescence imaging for optical imaging in small animals. But we believe that in the next 5 years, that will become the trend, and maybe from 5 to 10 years, that will be the standard. So today there are many groups over the world. I'm thinking of some groups in the USA, groups in France, in Europe, but also in Asia that are working on releasing commercially available probes for the NIR-II spectrum with different excitation from 800 to even 1000 nanometers excitation, and having emissions from 1100 to 1600 nanometers. So it's still the very beginning, but so far the most of the studies were done on ICG, indocyanine green, and we can see already a few commercialized probes or NIR-II markers available. So we believe in the next 5 years that won't be an issue to go from the NIR-I imaging to the NIR-II imaging.
Awesome. That kind of leads nicely into the next question, just around sort of where we see things going in the future. So Vilber is obviously at the forefront of all of these technologies in optical imaging. So what further advancements might we expect from technologies like the FT900? Do you think that this is going to be the most common optical imaging technology that people are using in 5, 10 years? Sort of what sorts of developments can we expect?
Yes, I think we can expect more and more commercially available probes for NIR-II. And in regards to Vilber, we won't stop to what we have done with the FT900. There are other developments in progress. Currently working on the AI, which is a common trend today. So the aim of the AI will be that whenever you start the acquisition, after the acquisition is over, your analysis will be already available just after the image is captured. So it will help researchers to save some time and quickly analyze, having the analysis already made for them. Also, you can still adjust the analysis if you need to, because the AI is not 100% sure, but at least it will be used to be a tool to make your life easier. So Vilber is already working on this, and that will be released as a future software upgrade, which will be free of charge. So that will be a bonus to the users.
Absolutely. One question just popped in there quickly around whether the webinar is recorded and available for future reference. Yes and yes. So I guess one last thing then, Alexi, what you just kind of described there, this is more maybe a comment than a question, but it is truly impressive just how much Vilber is able to offer in terms of customization with these products, right? So, being able to work one-on-one with us at Syntica as well as you guys over there, over in France to really make sure that the tools and the reagents and the different filters that a researcher might need. You guys are there to work with the customer to make sure that they are well equipped to get their research done. So I think that that's really impressive and was very much demonstrated by your presentation today. All
Thanks.
right.
Yes, indeed, we do have a standard FT900 instrument, but recently we've been asked to have it customized a little bit with other excitation sources and emission filters. So of course, that's something we can do. We can adapt our instrumentations to your imaging needs. So for instance, we've been asked for some excitation at 1064 nanometers. We simply did not put it as a standard because there are not many there are not many studies already available for some probes having an excitation at 10-16 nanometers, but since that specific customers wanted it for its own study, so we have made it available for him. And of course, we can customize other stuff, such as the emission filters, if you prefer more narrow bandpass filters or whatever you prefer. We are very versatile and flexible in regards to your imaging needs.
Absolutely. So lots of example images and videos to come. So that's great.
Yes.
All right. So if there's no other questions from the audience, we've reached the end of today's session. So again, to just be respectful of everyone's time, we'll wrap up here. As I mentioned at the start of the webinar, we'll be sure to answer any other outstanding questions in the written form of the transcript, and we'll get that out to you over the next few days. I'd like to thank Alexi again for his presentation today and for taking the time to answer everyone's questions. I trust that we've been able to provide you with some relevant information for the studies that you're currently working on back at home. If you have further questions about today's presentation or about any of the equipment that we offer at Cintiqa, including the Newton 7 FT900, I encourage you to reach out directly to us and we'd be happy to discuss things further with you. We welcome the opportunity to always review your specific research needs and how our equipment can help you move forward. So yeah, thanks again to all for taking the time out of your day to attend our session, and we look forward to seeing you at future Cintiqa events. Thanks and have a great day.
Thank you so much.
Thanks.
Merci.